Nervous system function depends on the flow of information through chains of neurons connected functionally, not physically, by synapses. The term synapse comes from a Greek word that means to clasp or join. Synapses mediate transfer of information from one neuron to another, or to an effector such as skeletal muscle, smooth muscle, cardiac muscle, or glands. The most common types of synapses are axodendritic, in which the synapse is between the axonal endings of one neuron and the dendrites of other neurons. The next most common are synapses between the axonal endings of one neuron and the cell bodies of other neurons. Less common types include synapses between dendrites called dendrodendritic, and synapses between dendrites and cell bodies called dendrosomatic. Recall that the neuron conducting impulses toward the synapse is the presynaptic neuron, and the neuron transmitting the electrical signal away from the synapse is the postsynaptic neuron. This means that the presynaptic neuron is the information sender, and the postsynaptic neuron is the information receiver. Most neurons function as both presynaptic and postsynaptic neurons. Neurons have anywhere from 1,000 to 10,000 axon terminals making synapses and are stimulated by an equal number of other neurons. There are two types of synapses depending on how the information is sent from the presynaptic cell to the postsynaptic cell. These are electrical synapses and chemical synapses. Electrical synapses are less common and correspond to the gap junction found between certain other types of cells in the body. They contain protein channels, made of connection subunits, that connect the cytoplasm of adjacent neurons and allow rapid and coordinated flow of ions and small molecules directly from one neuron to the next. Neurons joined this way are said to be electrically coupled, and transmission across these synapses is very rapid. In adults, electrical synapses are found in regions of the brain that are responsible for certain stereotyped movements such as the eyes. They are also found in the hippocampus, which is a region of the brain involved with emotions and memory. They are far more abundant in embryonic nervous tissue, where they help to guide developing neurons to their proper connecti. Chemical synapses are far more common than electrical synapses. Chemical synapses are specialized for release and reception of chemical neurotransmitters. A typical chemical synapse is shown above and is made up of two parts, one a knob-like axon terminal of the presynaptic neuron, which contains many tiny, membrane-bound synaptic vesicles each containing thousands of neurotransmitter molecules, and two a neurotransmitter receptor region on the membrane of the dendrite or the cell body of the postsynaptic neuron. The presynaptic and postsynaptic membranes are separated by a fluid-filled, tiny space about 1/millionth of an inch called the synaptic cleft. The above diagram summarizes the events that take place during information transfer across a chemical synapse. The arrival of the depolarization wave action potential opens voltage-gated calcium channels and allows calcium ions to flow into the axon terminal. This causes the synaptic vesicles to fuse with the presynaptic membrane, and neurotransmitter is exocytosed into the synaptic cleft. The neurotransmitter diffuses across the synaptic cleft and attaches to receptors on the postsynaptic membrane. Binding of neurotransmitter opens ion channels in the postsynaptic membrane, resulting in graded potentials in that membrane. The chemically gated ion channels open, leading to depolarization of the postsynaptic membrane. The neurotransmitter, in this case, acetylcholine, is destroyed by enzymes residing in the synaptic cleft, and or are taken back into the presynaptic terminal. This step is important for specific transmission because as long as neurotransmitter is bound to a postsynaptic receptor, it continues to affect membrane permeability and to block reception of additional signals from the presynaptic neuron. Depletion of neurotransmitter closes the ion channels and terminates the synaptic response. The effects of neurotransmitters generally last a few milliseconds before being terminated by one enzymatic degradation, reuptake by astrocytes or the presynaptic membrane, or can diffuse out of the cleft. Figure 11.12.2 Information processing involves interacting groups of neurons, and information is encoded in the frequency and pattern of action potentials. Many of the receptors present on postsynaptic membranes are specialized to open ion channels, thereby converting the chemical signal into an electrical signal. The neurotransmitters mediate graded potentials, local changes in membrane potential that are graded according to the amount of neurotransmitter released and the time it remains in the area. Postsynaptic membranes generally do not generate APS, axons with their voltage-gated channels do have this ability. Chemical synapses are either excitatory or inhibitory, depending on how they affect the membrane potential of the postsynaptic neuron. At excitatory synapses, neurotransmitter binding causes depolarization of the postsynaptic membrane. Instead of causing an action potential as occurs in axons, local graded depolarization events called excitatory postsynaptic potentials occur at excitatory postsynaptic membranes. Each excitatory postsynaptic potential EPSB lasts a few milliseconds and then the membrane returns to its resting potential. The only function of EPSBs is to help trigger an AP distally at the axon hillock of the postsynaptic neuron. If there is adequate excitatory stimulation, and currents of graded potentials are strong enough to depolarize the axon to threshold, axonal voltage-gated channels open and an AP is generated. Conversely, binding of neurotransmitters at inhibitory synapses reduces a postsynaptic neuron's ability to generate an AP. Most inhibitory neurotransmitters cause hyperpolarization of the postsynaptic membrane, making it more permeable to potassium or chloride ions. Sodium ion permeability is not affected. If potassium channels are opened, potassium moves out of the cell. If chloride channels are opened, then negative charged chloride ions move in. In either case, the charge on the inner face of the membrane becomes more negative, making it less likely to fire, generate an AP. A single EPSB cannot induce an AP. However, thousands of EPSBs coming from the axon terminals of many excitatory neurons are firing on the same postsynaptic membrane. The EPSBs can sum together to influence the activity of a postsynaptic neuron. Two types of summation occur. Temporal summation occurs when one or more presynaptic neurons transmit impulses in rapid-fire order and bursts of neurotransmitter are released in quick succession. Spatial summation occurs when the postsynaptic neuron is stimulated at the same time by a large number of terminals from the same, or different neurons. The individual EPSBs add up and result in depolarization. IPSBs can cancel out EPSBs, or can moderate the response because EPSBs and IPSBs mate all well. If the stimulatory effects of EPSBs dominate the membrane potential enough to reach threshold, the neuron will fire. If summation between IPSBs and EPSBs yields only subthreshold depolarization or hyperpolarization, the neuron will not generate an AP. Typically, each neuron receives input from hundreds to thousands of other neurons. This picture shows the cell body of a neuron covered with synaptic knobs purple of other neurons. The neuron will add up the excitatory input and the inhibitory input. If there is enough excitatory input to bring the postsynaptic neuron to threshold, then an action potential will be generated at the initial segment and propagated down the axon to the next neuron in the circuit. Neurons functioning groups, and each group contributes to broader neural functions. The nervous system is hierarchical. In order for there to be integration, the individual parts must be fused into a smoothly operating whole. I look at like having 1,000 hot cold water faucets, each with a slightly different temperature. Sometimes I'd like the bath water to be hot and steamy, other time cool and refreshing. To get the correct temperature output from the output faucet, I'd open various combinations of the individual faucets until I got the combination that yielded the correct temperature for the situation. In terms of the nervous system, the situation would be in response to stimuli leading to the appropriate output necessary to maintain homeostasis or to perform a voluntary action. Thus, millions of neurons in the CNS are organized into neuronal pools, which are functional groups that integrate incoming information received from receptors or different neuronal pools and forward the processed information to other destinations. An example of a simple neuronal pool is shown in the above diagram. Note that one incoming presynaptic fiber branches extensively as it enters the pool and then synapses with several different neurons in the pool. When the incoming fiber is excited, it will excite some postsynaptic neurons and facilitate others. Neurons most likely to generate impulses are those closely associated with the incoming fiber because they receive the bulk of the synaptic contacts. Those neurons are said to be in the discharge zone of the pool. Neurons farther from the center are not usually excited to threshold by EPSBs from the incoming fiber, but they are facilitated, meaning that they can be brought to threshold more easily by stimuli coming from another source. The periphery of the pool is the facilitated zone. Neurons in the pool are usually combinations of both excitatory and inhibitory neurons. There are 4 basic patterns of neuronal circuits. These are diverging circuits, converging circuits, reverberating or oscillating circuits, and parallel after discharge circuits. The above diagram shows an example of 2 of these circuits. A circuit, by the way, is the pattern of synaptic connections in a neuronal pool. Circuits determine the functional capability of the pool. In diverging circuits, one incoming fiber triggers responses in ever-increasing numbers of neurons farther and farther along the circuit. Thus, diverging circuits are often amplifying circuits. Diverging circuits are common in both sensory and motor systems. For example, impulses traveling from a single neuron of the brain can activate 100 or more motor neurons in the spinal cord and, consequently, thousands of skeletal muscle fibers. In a converging circuit, the pool receives inputs from several presynaptic neurons, and the circuit has a funneling, or concentrating effect. Incoming stimuli may converge from one area or from many different areas, which results in strong stimulation or inhibition. This explains how different sensory sensations can lead to a single type of efferent response. For example, the smell of a rose, listening to a beautiful song, a gentle caress, or viewing a breathtaking scene can all lead to a single motor output, a smile. In reverberating circuits, also known as oscillating circuits, the incoming signal travels through a chain of neurons, each of which makes collateral synapses with neurons in a previous part of the pathway. As a result of the positive feedback, the impulses reverberate are sent through the circuit again and again, giving continuous output signal until one neuron in the circuit fails to fire. Reverberating circuits are involved with rhythmic activities such as the sleep-wake cycle, breathing, and swinging your arms when walking. In parallel after-discharge circuits, the incoming fiber stimulates several neurons arranged in parallel arrays that eventually stimulate a common output cells. Impulses reach the output cell at different times, creating a burst of impulses called an after-discharge that lasts 15 milliseconds or more after the initial input has ended. Once all the neurons have fired, circuit activity ends. Parallel after-discharge circuits may be involved in complex, exacting types of mental processing. The above table summarizes the main points of information processing.